Hello, welcome to the last module of our course. These 4 weeks, we have discussed how to use ODEs, ordinary differential equation, to create mathematical model for biological system involving dynamics. Then we have discussed how to analyze those models. We have discussed how to use numerical simulation to solve those problems. And then we focused on particular type of model, for example, modeling cell signaling pathways involving positive feedback, negative feedback, etc., and also transcriptional network. In this last module, I will discuss about certain useful resources in the web which you may find very useful while creating mathematical models for biological systems, particularly for cellular models involving cell signaling, transcriptional network. So let us start. I will discuss on 3 specific topics for which you may have to look back into the internet to collect information. First one is to get information about the molecules and the rules involved in the processes. Suppose you want to create a mathematical model for a particular signal transduction pathway. For that, you need to know which are the molecules involved in this pathway. You have to know the cellular compartments where they are working. At the same time, you have to know who controls whom. That means you have to know the rule of the game. We often dig into the web to collect this information. Secondly, we may look into the web to look and find out old mathematical models. Old mathematical models are very useful thing to start our modeling. We can recycle existing model to fit our problem and answer specific question. So very often we look into existing model which are available in the net. And the third one is collecting information about parameters. If you remember, in an ordinary differential equation, I will have a dependent variable, I will have independent variable. At the same time, there will be some constant terms of the parameter values. For example, in a enzymatic reaction, I have to know the rate constant, which are the parameter in the process. So I have to know the numerical value for these rate constants or parameters. Where do I get those parameter values? Often we go back to the web and use different web resources to collect those information. I will start discussing with collecting information about molecules and pathways or the components of the model. Every model, whether it is cellular level or at a higher level, are made up of multiple components. At cellular level, these components are essentially molecules involved in pathways and processes. Where do I get the information about all these components which are relevant for my modeling? The best one you have to always remember is digging into existing literature. There is no other alternative to it. We have to collect all relevant publications and we have to rigorously read and distill information from those publications. And one web resource which comes very handy for this purpose is obviously PubMed, the screenshot of which I have shown here. PubMed is a repository of abstracts of our publication over the ages, and it also connects to the publication, the paper, if it is available freely. And most of us who work do research in biology are well acquainted and use PubMed regularly. Some of us prefer Google Scholar to dig into the published literature and find out relevant paper. Whatever resource you use, ultimately, if you want to create a successful model, you have to read and distill from these published papers. Now, when we are trying to create mathematical models for cell signaling pathway or transcriptional network, usually these pathways and networks involve multiple components, and it becomes sometimes a bit confusing to distill from literature and then create the model. And to help you in this purpose, there are certain databases which actually curate manually information about different pathways. I have listed some of them here. For example, one is KEGG database. I use it regularly. Then there is a Reactome, which is also very rich curated database. Then you have Panther, wiki pathways, and many more. I have just listed 4 here, and I will discuss KEGG database in details because I find it very useful in day-to-day work on modeling cell signaling. Let us look into KEGG database. I have given the web link here. If you go to this page, you will find lots of information there for pathways. Remember, KEGG is a manually curated database. That means some people are reading through the published literature and distilling out information and drawing the pathway diagrams and curating that information in an organized fashion. You as a user can search through this database and get relevant information. For example, suppose I want to know all the molecular pathway in which the molecule AKT, which is a kinase, is involved. So I can write AKT in this search text box and then click go so that I get a comprehensive result of all pathways where AKT is involved. And the result will be like this. In a new page, you will get a table where comprehensively all the pathways in which AKT is involved as per the KEGG database will be shown. You can look into this. For example, this one is a particular pathway. For example, written here, it is PI3K AKT signaling pathway. If you click on it, it will open up and you will get an expanded view of the pathway. I will show you separately. The other way of searching is that from the main page, you notice there is something called genes. So I can search for a particular gene. So if you click on that, you will get a new window. So where you can type the name of the molecule. For example, I am typed here AKT, and you can also type the name of the organism. For example, I have written HSA, that is a human. And then if you search by clicking this go, you will get a detailed list of search result. Here in this window, all the places in the database where AKT is present is listed. So it has listed all other molecules which are close to AKT or has similar name. If you find here, this is what we are looking for. This is the entry which we are looking for because we are looking for AKT or the synonym of that is PKB. So if I click on this entry, it will give me a list where I will have detailed information about this molecule as well as I will have information about the pathways in which these molecules are involved. So here it has listed all the pathways in which the AKT molecule is involved. And suppose I am interested to know the PI3K AKT signaling pathway. So I will click on this particular entry for pathway so that I can get a view of the pathway. So let us see what will happen if I click on that. If I click on that, a pathway map will appear. And you can easily see I have AKT here in red. If I zoom, you can see what we have here. I have AKT, that the molecule I am looking for. The map is saying if I give a growth factor GF that activate RTK, that is receptor tyrosine kinases, a particular group of receptor, that will activate IRS1, a intermediate molecule, that in turn will activate PI3K, that will activate or rather produce PIP3, a lipid, which activates by shown by this arrow by phosphorylating AKT. Once AKT is active, it can inhibit GSK3 by phosphorylation. This plus sign means phosphorylation. This hammerhead means inhibition. So active PAKT will phosphorylate P21 and inhibit this one. And in this way, you can easily see which molecule is controlling which molecule and how the control works, either through phosphorylation, dephosphorylation, or through transcriptional process, like if the molecule is a transcription factor. So in this way, you can search through KEGG database and collect information about different molecules involved in a pathway and how they are connected with each other. So whatever information you collect from literature gets supplemented by the information present in the KEGG database. Now let us go to the second issue. If you remember, the second issue for which we will prefer to go back to the web and search through it is for old models. Suppose I am creating a new model for a particular signal transduction pathway, say for insulin signaling in glucose metabolism. I have to write down some ODE representing for this model. It will be very helpful if I look into the literature where somebody else has previously created a similar model and I try to use that type of equation so in my model. I sometimes may prefer to collect information about parameter values used in those models. I may sometimes borrow the whole model and tweak here and there and create a new model. That means I want to recycle an existing model. For this, I have to go back to old models. One way of going to old models is look into the literature. But nowadays, there are many databases which has come up where scientists store their model, they submit their model which they have published in a paper, and those models are manually curated and shared through the database. So anybody can go back and search those model databases and retrieve the existing model and use it for their own purposes. Many such databases exist. I have listed few databases here. For example, one is biomodel database. I will discuss about it a bit more. Then you have CellML database. And then JSIM web page, if you remember, it has an archive of models. So if you go to the JSIM web page, you will find an archive of models where you will find lots of models, not just based on JSIM tool, but developed by other groups using other tools, but available there in that archive. And you can actually retrieve those models and run in JSIM and play with them. So let us look into biomodel database. If you go to the biomodel database, there is a large page, and the most useful part for us is this search window where you can directly put an entry. For example, I have written AKT here because I want to search all models submitted and curated in database which involve AKT. So if I do that, I get a search result which gives me a long list which I have truncated here and shown, a long list of all curated model where AKT is involved. So for example, suppose we want to see in detail about this model which involves NGF, that is nerve growth factor, and EGF signaling. So if I click on this biomodel entry, this is the biomodel ID for this model, I will get detailed information about this model. So what information I get? I have detailed information about the people who created the model, publication where it has been published, and all others. My focus would be primarily on this download option, overview, math, physical entities, parameters, these tabs. These information present in these separate tabs are very useful. For example, I may want to download this model and simulate it myself in my computer. Now remember, there are not only one single modeling framework. If you are using ordinary differential equation to create model, you can use different tools to create that model. We have extensively used JSIM. Somebody may be using C, somebody using maybe Fortran, somebody may be creating MATLAB, using MATLAB to create their model. Now the issue is then how will we share these models? One way of sharing this model is to convert them in a single language and then share it. And that single language is systems biology markup language. So all models submitted in this biomodel database are actually converted into model made up of systems biology markup language. And you can download that model in SBML language. And then you can export that to your software. For example, I can download the model from this database for this particular NGF and EGF signaling by clicking here, download SBML, and you see I get a multiple option to download the same model in SBML format. So suppose for example I can download this one, SBML L2 version 1 curated, that means that it has been manually corrected and curated. So if I download that file, I will get a model written in SBML. And if you remember, actually JSIM can read a SBML file, and then it can be simulated using JSIM. So I can download this one and then open it in JSIM and simulate myself, and then I can manipulate if I require. It's very useful. Let us look into the other important tab. For example, the math tab, it lists all the unit processes involved and the mathematical reactions involved there. For example, EGF binding is represented by this rate constant into EGF into free EGF receptor. So all the mathematical formulation which are used to create the ordinary differential equation representing rate of change of some molecules are listed in this tab, math tab. Then you have this parameter tab. If you click on this parameter tab, what you get? You get numerical values of all parameters used in this model. For example, this one are all kinetic parameter constant values used for this model. So this tab is very useful. For example, if you are looking for a parameter value for a particular process in your model and you are not getting that information, you can dig into biomodel database, and if you find a suitable similar model, you may find out the parameter value from this particular tab in a model. So in this way, I can get the model, information, the mathematical equation that makes the model, the parameter values, and as a whole I can download the model in a SBML format and use it as per my use purposes. Let us come to the third issue, for which we may want to go into the internet and use different web resources. That is to get the parameter values. So to run a model, to simulate it numerically, I require the numerical value of the parameters, the constants. Where do I get them? Obviously, the first option is to look into published literature. So suppose you are creating a mathematical model where you have an enzyme which catalyzes a particular phosphorylation of a protein, and if that process is Michaelis-Menten, following Michaelis-Menten kinetics, you may look into old literature to find out if somebody has measured the Michaelis-Menten constant for that reaction. If you are lucky, you may get that information in literature and you may use those numerical value in your model. But most of the time, this type of value, numerical value for a particular process or particular kinetic constant are not available. In that case, many a time we go back to old models dealing with similar processes, and we use some educated guess to use those parameter value which other people has used. For example, if you are modeling a particular single transaction pathway where a kinase phosphorylate a particular protein Y to phosphorylated Y, you have not got the kinetic rate constant for this phosphorylation reaction. But you have seen another model where people have estimated experimentally the kinetic constant for a similar reaction but for a different kinase and substrate. But you have a reason to believe that your kinase and substrate will also behave in the same fashion. So you may reasonably take that numerical value used in that old model for your modeling. And very often modelers do that. The third option is obviously, if you can afford to do, is do your experiment yourself, and from those experimental data you estimate the value of parameters that you have to use for modeling. I will discuss that in one slide. The other and the fourth option, where we may go back to the web to collect information, is there are certain databases which stores this type of data collected from literature and helps you to find those numerical values a bit easily. Let us first look into parameter estimation from experimental data. Sometimes people prefer to do their experiments themselves and generate experimental data in different experimental conditions. From that data, they want to estimate or generate their parameter value for their model. How do they do that? It involves multiple steps. Let us look into the steps. First, you perform organized experiments. Then you create a mathematical model using the ordinary differential equation that will have dependent, independent variables and parameters which you do not know. Their numerical values you do not know. Then you systematically try to use different parameter values. That means for each parameter you systematically take different values and simulate your model and try to fit your simulated data with experimental result. So what you are doing, suppose your ODE has two parameters. You have a model using one single ODE and you have two parameters K1 and K2. You assume that K1 varies from 1 to 10 and K2 varies from 1 to 10. Take different combination of K1 and K2 in this value range and then simulate your model. The simulated model will give you some output, some result. Now try to see how different those results are from the experimental result. And try to find out the parameter value which gives you best fit. That means which gives you best simulation output which is closest to the experimental one. So it is an iterative process and depends upon how systematically you search the parameter space to find out the optimum parameter value, and it usually takes long period of time to do that. And once you have identified the parameter value which gives the best fit, you use that for other purposes. There are many ready-made tools for this type of parameter estimation, but you have to remember no parameter estimation algorithm can give you 100% assurance that you will always get the best parameter estimate. You have to try and do trial and error to find out which one is working good for your particular type of problem. JSIM itself has an option to do parameter estimation. For example, as I have shown in this figure here, if you notice, there is a tab in JSIM called optimization. If you click on that optimization tab, you can actually run a parameter optimization algorithm to find out the best value of parameter which generates simulated data which fits best with your experimental data. If you want to learn it more, we have not discussed this part of parameter estimation in our course. If you want to learn it more, you may look into the JSIM web page. You will find lots of information about this optimization. Then many other ready-made plug and play software like Copasi for modeling has this parameter optimization option. Then there is another tool called D2D, Data to Dynamics, which is written in MATLAB and you require MATLAB to run it, but it is quite a powerful tool which gives you lots of freedom and lots of different algorithm to do parameter estimation. Now suppose you have done parameter estimation or have collected information from the old models, but it will be always good if we have a database where numerical value of different constant, kinetic constant or different rate values or other biological values are stored. There are a couple of databases which are for these purposes. One of them is BioNumber, which is an extensive database and we use it very frequently. Then you have Brenda, which can curate information on enzyme kinetic data. Then you have SABioRK, which stores reaction kinetic data in its database. I will discuss a bit about BioNumbers. The advantage with BioNumbers is that BioNumber stores lots of different types of data. For example, you may need to know the size or an average size of a mammalian cell. You can go to BioNumber to search it. Suppose you need to know what is the average number of insulin receptor present on a human cell. You can dig into BioNumbers to get that data. So it's not just the parameter value, for example, rate constant that you used in your kinetic model, but other numerical values which are very useful in our modeling are deposited and curated in BioNumbers. So modelers very frequently go and dig information in BioNumbers. Let us jot down the key points. The main source of information for building model is always published literature. There is no other alternative to it. Databases like KEGG help us by providing information in a curated format collected from this published literature. Published or old models are good starting point for creating a new model. These models provide information about the system, that means which are the molecules involved in the pathway, the mechanism of action of different molecules. You get this information from old models. This old model help us to make mathematical equation, that means it helps us to write down the mathematical model, and it also provides us estimate of different parameter values. There are certain model databases where the existing model or published models are stored. For example, one useful one is biomodel. These biomodel databases are for storing, sharing, and for recycle of published models. Parameter values that are required for numerical simulation of any ODE based model, you may collect them from literature, old model, or by parameter estimation through your own experiments, and sometime you may get those values from numerical databases which store biological numbers. That's all for this module. We are at the end of our course. Thank you for being with us.